All right, so the last lecture that we left off on was on central dogma. We briefly touched on transcription, translation, and DNA replication. So we're going to use this unit to try and dive a little bit deeper into each of those concepts to better understand the intricacies of central dogma. And today's focus will be on DNA replication. Because DNA replication is such a critical part of normal biology, there's typically not any disease where life is sustainable where DNA replication is dysfunctional. And so instead of that, what we're going to focus on is instances where there's an outside trigger that alters or affects DNA replication. And the focus here today is going to be on chemical and biological warfare. So the use of chemical and biological weapons has an unfortunate and long history, dating back to 429 BC, where during the Peloponnesian War, there were toxic fumes that were used by the Spartans back in the between the 10th century to the 13th century. The Sung Dynasty would use arsenic as a tool in battle. In addition to that, the Mongols would also use contaminated corpses that were infected with the plague and use those as a weapon during battle. And there's also, beyond that, lots of modern instances as well where chemical and biological weapons have been unfortunately used. The focus of the chemical weapon that we're going to focus on today is going to be mustard gas. It is a powerful alkylating agent. What that means is that it adds an alkyl chain as a modification. Typically, mustard gas can be inhaled, and because of that, it's going to be targeting a lot of the mucous membranes from the eyes to the respiratory tract, as well as and what's also key is that it's a vesicant, meaning that it's a blistering agent. What's unfortunate also is that it can be easily made and dispersed. I have a hunch, I don't know if you all remember, it's so easily made that often that it can be a concern for some orgo-chem labs that it can actually be easily created there. And I have a hunch that quite possibly with the chemistry building issue where the FBI came on grounds, that this could have been what might have been at play. It wasn't. It was a bomb? Okay. Sorry, getting back on track because I have like 10 more questions, but that was my hunch, but all right. What's also true about mustard gas is it can be stored for a long time. In addition to that, because it is released into the air, it can be difficult to treat and decontaminate. And in addition to that, difficult to protect against. Just as a heads up, there's going to be images demonstrating how it's a blistering agent and how it affects some of the mucous membranes. And so it can cause erythema, which is inflammation of the mucous membranes, conjunctivitis, which is inflammation of the conjunctiva within the eye. It causes lots of blistering, burning, lots of irritation within the nasal cavity, which causes sneezing and nosebleeds. In addition to that, it can cause lots of coughing, pretty severe sore throats. As it gets more severe, it can cause pretty severe vomiting. The LD50, does anyone want to define LD50 for us? Mm-hmm.
It's like the dosage for
Yeah, half of a population. Yeah, exactly. It's LD50 is a lethal dose at which 50% of a population would die. And so here it's around 1,400 milligrams. What's important to note is that when it's inhaled into the lungs, the LD50 is only about 10 milligrams there. And so based on the really vast surface area within the lung, what's key to note is that the severe effects there can be noted around 500 milligrams per minute per cubic meter there. Another biological weapon that we're going to discuss today is anthrax. It's historically called wool sorter's disease, but I'll actually talk about why that is in just a moment. It's derived from the bacterium Bacillus anthracis, common soil bacterium. It's primarily infecting large vertebrates like sheep, based on the name wool sorters disease. In unfavorable conditions, it can form a spore, and so when oftentimes for some bacterial species, whether that's based on temperature, pH, or any other environmental condition, when there's unfavorable conditions like that, some of these bacteria, in this case, Bacillus anthracis does so, it creates a spore to protect itself until it is within a more favorable condition where it can then grow and become fairly infectious. The key things to note here about the spore form of anthrax is that it can actually be stored for a really long time. It can easily grow, easily disperse, has a fairly high mortality rate, and is non-transmissible, meaning that if someone is infected with anthrax, it's typically not going to be spread to others. Before most of you, I think, were born, right before 9/11, there was an anthrax scare in the DC area where federal employees were sent anthrax spores and received anthrax spores in their mail. And so there was a long time where there was a fear of anthrax in the mail. And so this is, again, because of a lot of these features that we just ran through, it's oftentimes used as a weapon. One of the most common manifestations of anthrax infections is cutaneous anthrax, or an anthrax infection in the skin. It often occurs when there's a cut or abrasion that's been exposed to the bacterium. So the reason why it's historically called wool sorters disease is, as I said earlier, it primarily emerges in larger vertebrates. One common one is sheep. And so, a couple centuries ago, typically when wool sorters, the people who would use shears to cut the wool off of sheep, oftentimes that's an occupation where there's lots of nicks and abrasions on their skin. Wool sorters, the people whose job it was to collect wool, would oftentimes be exposed and suffer from cutaneous anthrax infections. And so that's how it got its name. As long as antibiotic treatment is available, it's not normally lethal. Oftentimes, it's as depicted here, it can emerge as a raised bump with a painless ulcer, as well as what we call a necrotic center. So we've talked about apoptosis as this programmed cell death that's really well regulated. Necrosis is unprogrammed cell death, massive cell death that's not regulated at all. I'm sure some of you have heard and seen instances of something called like necrotizing fasciitis. It's just like large amounts of tissue death, cell death that's not regulated. And so here you have a necrotic center present at this at this ulcer. In addition, you also get swollen lymph nodes. We've talked about already how lymph nodes are a secondary lymphoid organ. And so when there's an infection like this, there's going to be activation of the immune system, which oftentimes leads to the expansion and growth of T cells and B cells within the local lymph nodes there. So that's cutaneous anthrax. Inhalation anthrax is when the spores are inhaled. So it begins in the lungs and incubates over the course of several days, up to a week. Once symptoms emerge, that's when patients typically have only 1 to 2 days to live. There's approximately a 100% mortality rate after 1 to 2 days after symptoms emerge, after this incubation period. That's if it's left untreated. If patients are treated with strong antibiotics like Cipro, oftentimes there's a less than 30% mortality rate. The lethal dose there is around 10,000 spores in the lung for humans. The symptoms begin as a fairly, as a fairly common appearing upper respiratory infection. After a few days, then it'll advance to dyspnea, shortness of breath, diaphoresis, which is excessive sweating, myalgia, which is muscle pain, and then eventually lead to septic shock. So we've talked about shock as a general phenomenon where there's a drop in body temperature, severe drop in blood pressure, such a dramatic drop in blood pressure that organs are not able to sufficiently be perfused with blood, and you undergo organ failure. And that's typically how people die from shock. and there's different forms of shock. So here, septic shock is a result of an infection that went from being localized to now systemic. So when you hear someone has gone septic or they're in sepsis, that's where an infection can go. Once it hits the bloodstream, typically that person has become septic. So a lot of times, I'm sure a lot of us have all had strep throat at some point or another. It's really important to get your antibiotics because strep throat and a fair number of people can actually lead to sepsis. It can go systemic. Once it hits the bloodstream, that's when a lot of problems can occur. And that's the final phase of inhalation anthrax here. So why are we talking about mustard gas and anthrax? What's key to note is that mustard gas is noteworthy because it inhibits our own DNA's ability to replicate. It affects our own DNA replication machinery. Anthrax, the way in which it's treated is by using drugs that help inhibit bacterial replication. So the common denominator here for both of these is DNA replication. Before we move on, are there any questions here? Yeah. I have watched the show. I never saw that one, unfortunately. I'll make that my homework for tonight. Are there any other questions? Yes. I don't know if that's why they do it, but oftentimes there have been a few studies that have demonstrated that UV light is not great as a sterilization technique. That might very much be why, but typically, like even in a research lab, our cell culture hoods have UV light, and it's been shown a lot of times that it's not great at disinfecting those surfaces, and so that could be why they're doing it, but it might not be the most effective way to try and target anthrax spores or any sort of like infectious agent. Yeah. All right, let's go ahead and press on. So we one of the key things to note here, we're going to go ahead and dive into DNA structure here. One of the key things to note here is that our DNA is made up of 4 key base pairs. We have adenosine, thymine, guanosine, and cytosine, and that we have the base pairing of A to T and G to C. The way in which that base pairing happens is via hydrogen bonding that occurs between them. And what's key to note here is that we have our the base pair abbreviation in the center here. And so what's key to note here is that between our adenosine and thymine pair, we have 2 hydrogen bonds, and between guanine and cytosine, we have 3 hydrogen bonds. And so what's key to note is that this helps with the specificity of our DNA, whereby there's specific bonding that is meant to occur between these certain base pairs, and that kind of drives why we have A to T and G to C base pair matching. And that helps drive the complementary nature of our DNA. And so what I should have started with, forgive me, is that there's 2 key features of DNA that I want you guys to take away for this slide with, that our DNA is complementary, and that's because of what we just described. The next thing is that it's anti-parallel. What that means is that similar to our protein where we have directionality, we have an N-terminus and C-terminus, here we have directionality of 5' and 3' ends of our DNA. And so one end is going to be, we have on the left-hand side, we have 5' at the bottom and 3' at the top. On the right-hand side, it is opposite. It's 5' at the top and 3' at the bottom. And so that is what we call the anti-parallel quality of our DNA. And so during replication, one of the key enzymes at play is DNA polymerase, as we talked about during the central dogma lecture. And so the complementary nature of our DNA, DNA polymerase uses that to read what we call the template strand. We talked about how there's a specific, during replication, the 2 strands are separated, and DNA polymerase comes in and will read one of the strands as the template strand to make a complementary strand as a result. There are a few key things to note about DNA polymerase, and this is going to become more clear as we dive further into replication. There's a few key things that DNA polymerase cannot do on its own. One key thing is that DNA polymerase cannot melt or separate the DNA on its own. And so DNA polymerase needs another molecule to come in to melt or separate the DNA strands to enable replication to happen. Another really, really important thing to note is that DNA polymerase can only elongate a pre-existing strand. So we have our template strand, and there's going to be the new strand that emerges. For this new strand, DNA polymerase cannot initiate a new strand on its own. There needs to be something there for DNA polymerase to elongate. So it can't initiate, it can only elongate for the growing new strand that's going to be complementary and anti-parallel to the template strand. And what's really important to note here is that DNA polymerase can only elongate the new growing strand in a certain direction. It can only elongate in the 5' to 3' direction. Okay, so that means, and again, I'm going to try and make sure I have I'm as clear as possible with what strands we're referencing. When DNA polymerase is making a new strand, it's only in the 5' to 3' direction, which means it's reading the template strand in the 3' to 5' direction. Okay, it's going to be important to kind of have a reference point of what strands we're talking about. So the new growing strand that's going to elongate is created or synthesized in a 5' to 3' direction by DNA polymerase. And so there's going to be we're going to circle back to this inability of DNA polymerase to initiate, the inability to initiate and the inability to melt the strands. So like put a mental asterisk there because we're going to circle back to that. The sites at which replication occurs, or where it begins, where it's initiated, is at these sites called origins of replication or replication origins. Either phrases are fine here. And there's a specific class of proteins that help identify these replication origin sites. And these proteins are origin binding proteins. And throughout the course of the lecture, starting from this slide, we're going to distinguish between prokaryotic or bacterial DNA versus eukaryotic DNA or mammalian DNA. And so we're gonna have lots of points where we're going to be distinguishing between the two because there are different proteins that serve each of these different functions, as well as different structures of our genome here. What's important to note is that these replication origins might contain specific DNA segments with multiple short repeats here. When I say may contain, it depends on what system we're talking about with eukaryotic versus prokaryotic. What's important to note is that in the prokaryotic system, these DNA segments are well known and characterized and called consensus sequences. Those are the specific sequences that have been identified as the sites at which it is a origin of replication. That's what's depicted in the top half in green. That's what I see as green, so forgive me again, I'm colorblind. So I'm going to call that green. So that is an identified consensus sequence within prokaryotic cells. Within eukaryotic cells, it's less well known where these origins of replication are, but what is known is that they are particularly rich in AT sequences. Does anyone want to venture a guess why the site of replicate or these origins of replication in eukaryotic cells would be rich in AT base pairs? Exactly right. Because there's only 2 hydrogen bonds for it to break. So there's a lower threshold to separate there, as opposed to if they were GC rich locations. And so the bottom half with the red sequences, there's going to be multiple sites where replication can be initiated within eukaryotic cells. And so we don't have specific sequences here that we know, but we know, as depicted here, that they're rich in these AT base pairing. What's also important to note is that in some of these segments here, there's going to be you'll see N depicted there as depicted up in the top left box here. N is just going to denote for any nucleotide. That's all that means there. So within the prokaryotic system, the way in which this is just to help you understand how we determine what the consensus sequences are. Excuse me. This was determined bioinformatically, which means that these sequences were all the genomes were all lined up across different prokaryotic species, and the common overlap at these origins of replication helped us identify what the specific sequences are that are conserved across prokaryotes. So we have here to the left the prokaryotic genome, which is a circular genome, and to the right we have eukaryotic DNA that's packaged in chromosomes, but here we're depicting it as linear DNA, imagining that it's been totally unraveled and we just have one long string of our DNA that's not packaged as it typically is. And so again, the prokaryotic DNA has specific identified consensus sequences. Our eukaryotic DNA doesn't have these well-characterized segments, but again, there are sites at which replication is initiated, these origins of replication. So typically there's one specific origin of replication for prokaryotic DNA right here. And from there, there's replication that occurs in both directions, creating this bubble right here. Where we have the parent strand up here and the daughter strand or the new strand of DNA emerging there. And when replication, when the polymerase is done elongating, you'll have two complete circular genomes, but are linked together almost like magician's hoops that are kind of stuck together. And so there's going to be a point at which that needs to be separated. And so that's going to be something we'll talk about for further in the lecture. For our eukaryotic DNA, there's going to be multiple sites at which replication is occurring. There's going to be multiple origins of replication. And so each site at which it's happening, you'll have the formation of what's called these replication bubbles. And the ends of those bubbles are called replication forks. That's going to be helpful further down the road as we help try and orient ourselves with the growing strand in which direction it's growing. And so the key thing to take away here is that we have very specific structures that I want you to take note of, these replication bubbles, where at the end of them, there are these replication forks. And there's going to be multiple sites at which replication is occurring within our genome. And it's going to eventually reach a point where those growing strands of DNA are going to all come together and fuse to create two now separate complete strands of DNA. And another key thing I want you to note here is that this is, again, happening all along the entire chromosome. I'm going to go here and then there. Go ahead. Yeah, so consensus sequences are these like identified segments of DNA, these identified segments of DNA. And so that's only been identified in prokaryotes. In eukaryotic DNA, it's less well known. We don't really know like what these like specific like down to the nucleotide level, what they all are. But we do know that a quality that they have is being rich in AT base pairs. Does that tackle it? Okay, go ahead. I'm going to correct your question really quick. So again, to kind of go off of his question about consensus sequences, they're only, for the sake of this class, we're only going to use the term consensus sequence to describe the replication origins in prokaryotic DNA. In eukaryotic DNA, we don't have identified segments known down to the nucleotide level. So consensus sequences, by definition, are known segments of DNA. We don't know down to the nucleotide level for eukaryotic DNA, but we do know that these replication origins in eukaryotes are rich in AT base pairs. Does that help answer your question? Great. Go ahead. Yeah, so the replication fork is all it is as the new strand of DNA is growing, we would refer to the space between the two as like, you can almost call it a replication bubble. It's not as it's not as easy to appreciate as it is with eukaryotic DNA, where like you can see a distinct bubble there. But the replication forks are basically just the end at which like replication is actively happening. Because as you can probably envision, replication is happening in that direction over here, and replication is actively happening in this direction. So an easy way to think about like the functional importance of a replication fork is that it is the site at which replication is actively happening and pushing out to further ends of the DNA. Does that help clarify? And it's also going to make a little more sense in about 10-ish slides when we talk about why the replication fork is important. Yes, last question. Great question. It's just the daughter strands. So for the sake of the recording, the question was, is the replication bubble comprised only of daughter strands? So what I should say is, forgive me, as it's depicted here, it looks like it's just a daughter strand. It's important to note that, as I was just answering to the previous question, the replication forks are sites at which replication is actively happening. And so the presence of the template strand is really important for the continuance of replication and the growing of the replication bubble towards another one. And so the daughter strand, by definition, there are daughter strands on the inside there that make up the replication bubble, but the availability of the template strand for replication to happen is also really important. All right. We're going to press on, and as we keep moving along, I think some of these questions are going to help be clarified a little bit further. So we talked about how DNA polymerase cannot initiate on its own, so it needs a protein that serves as an initiator. So here we have what's called an initiator protein. So it binds to the origin of replication, and it may separate the strands. Again, every time you see may, that's going to be a point at which we draw a distinction between prokaryotic and eukaryotic. But the key thing to take away from this slide is that to meet the need of DNA polymerase not being able to initiate, we have initiator proteins that bind to the origin of replication, and one class will separate and one class won't. And so we have here a class of initiator proteins. We have DNA A for prokaryotes, which is able to separate or melt the DNA. In eukaryotes, we have origin replication complex, or ORC. It's this hexameric protein structure that is not able to separate strands. So the prokaryotic DNA A can separate the strands. The ORC complex cannot. And so we're going to need a protein that's able to melt or separate the DNA strands here. One thing to note is that this is a barrel and ribbon diagram, and what we have here is that the barrels depict alpha helices here. Yes. It is really important for identifying the origin of replication. DNA polymerase, there's all of these proteins that we're going to talk about today, each serve an important function to help recruit and bring in, promote the activity of one another. So, for example, the ORC complex cannot separate the strands. There's going to be another protein we talk about that is called helicase. That is what in eukaryotes melts and separates the strands, but ORC directs where the DNA needs to be melted and separated. You don't want helicase, this protein that melts the DNA, to act indiscriminately throughout our DNA. You want a more targeted approach. And so ORC directs where helicase will bind to melt and separate the DNA. Does that help clarify? Great. That was a great question. So again, just to repeat for the sake of recording, helicase is the protein that will promote the separating or the melting of our DNA. This is an energy intensive process, so this requires energy. It means, and whenever you see requires energy throughout the course of this lecture, that means it's going to be hydrolyzing an ATP. And so there's going to be an ATP spent that promotes the melting or separating of the DNA. We have two classes of helicases. We have a prokaryotic and eukaryotic. DNA-B is the prokaryotic helicase, and MCM, which is the mini chromosome, should be mini chromosome maintenance complex, if I remember correctly. Yes, mini chromosome maintenance complex. It's again this hexameric protein structure that will help unwind. And I know it says below unwind single-stranded DNA. It unwinds to create single-stranded DNA, and so that's the naming, the nomenclature for these different helicases, as well as how they function. So just to summarize, we have helicase that melts. Oh, sorry, do you have a question? Yeah, so that's a great question. So the question is, why do prokaryotes have a helicase if DNA already can bind and separate the DNA? My hunch is that because DNA replication is such a key process in how in cell biology and cell functioning, that DNA-A might not be sufficiently good at separating or melting DNA, which required DNA-B to over time. There's an evolutionary need for a actual prokaryotic helicase to be formed, for lack of a better phrase. And so I think it's a great question, and so I'm for questions like that, I'm trying to think about them within the context of like evolutionary constraints, and I suspect that there needed to be a more professional helicase even for prokaryotes, despite DNA being able to do the same, being able to do it as well. Yes.
For the prokaryotic versus eukaryotic helicases, it says like that they're unwinded to see if the DNA is 530 or 0.5.
Yeah.
Why is that? And then also, are those the template strands?
Yes, it's only the template strand because at the point of helicase function, there's no new strand yet. So that's only the template strand. And your question is, why is that the case?
Yeah, why are they?
That's a good question that I don't know the answer to, why they have different polarity. I will look into that and get back to you. That's a great question. Okay, so once we have melted separated DNA, one thing that's important to note is that it's fairly energetically favorable for the melted DNA to reanneal or to come back together. So you need some sort of protein. You need something else to come in to help stabilize and keep the strands from reannealing. And so that's what we have, the single-stranded binding proteins, or the SSBs. They help ensure and stabilize melted DNA so that they don't reanneal back together. What's important to note is that when DNA is separated, there's oftentimes a problem on the DNA at the ends once they're separated. There's this issue of supercoiling. That might be hard to appreciate just by word of mouth. So I want to try and use an illustration. Now, can I get your help here? So given the fact that there's a big age gap between y'all and me, this is a phone cord that y'all might not have seen before. Sorry, I'm giving you a hard time. It's easy, I'm sorry. But basically, the idea is we have our double-stranded DNA that is wound in a helix form. Can you hold both ends for me? So if my hands are the going to the origin of replication to separate the DNA, when I pull it apart, and usually what's important to know is that when the DNA is melted, it's not just like a small region, it opens up a lot. And so if you look at the ends close to Najwa's hands here, there's the amount of coiling has gone up dramatically. And so that can be really problematic for the DNA. So we need a class of proteins to help address that supercoiling so that it doesn't create problems, structural problems for the DNA. Thank you, appreciate it. So we have a class of proteins called gyrases that help eliminate that supercoiling that's happening as a result of the DNA being melted and separated. This class, yes.
Were all three of those proteins initiator proteins?
No, great question. So the initiator proteins, so we talked about two of them, one for prokaryotes and one for eukaryotes. DNA A is a initiator protein for prokaryotes, and it's ORC complex or ORC for eukaryotes. So they go in and they identify the origin of replication. DNA A can separate, but we also have helicases that are who are professionally tasked with separating the DNA. And once it's separated, the SSBs, single-stranded binding proteins, actually come in and help stabilize it. I'm going to go ahead and ask. I'm going to ask for some volunteers to help do the same thing again, but now have a role for each of these proteins. Do you mind joining me up front and help being an initiator protein for me? Najwa, can you help hold the DNA strand again? Do I have a helicase out there? Come on up. You want to wear this one? Do I have a single-stranded binding protein that wants to join us? Come on up.
Thank you.
Okay. So to kind of help dramatize the entire process, but to kind of see everything working together. Okay. So we have our origin of replication that's somewhere along this double-stranded DNA here, this double helix. So our initiator protein comes in and identifies the specific origin of replication. So can you come in and identify just by grabbing onto it? There we go. So assuming we're in eukaryotic DNA here, our initiator protein, does anyone remember what the name of eukaryotic initiator protein is? ORC. Way to be self-aware. So because ORC cannot separate the DNA in and of itself, we have our helicase that will come in and see the initiator protein, see ORC, and separate the DNA there. So you can go ahead and pull it apart. Because our DNA wants to re-anneal, we have our single-stranded binding proteins that come in and dock on, you can come over on this side, and just basically dock on sides over there. And if we had another set of single-stranded binding proteins, they'd be on the other side of the replication bubble, helping to stabilize. All right, thanks, guys. I appreciate it. Round of applause for our folks. Thank you. Okay. You can keep your labels. Yes. We're going to get there. That's the next set of slides we're about to talk about. I'm going to go here and then here. Yes. Great question. So the question is, are all three of these, I'll actually, on this slide, I'll replace, for the sake of what we're discussing, replace gyrase with the initiator protein. Are all three, just because we have not really talked about gyrase yet, only in relation to its role as supercoiling, but initiator proteins, helicases, and single-stranded binding proteins. You asked, are they present in for both prokaryotic and eukaryotic? Yes, they are. For the helicases and initiator proteins, they go by different names, which is important to note. And what their different names are is important for you guys to know. There was another hand a moment ago. Was it your hand? Great. Okay. All right. So where we left off are these gyrases, these these class of proteins that are meant to help address the supercoiling that happens as a result of DNA being melted and needing to address that, overcome that hurdle. So there's an entire class of proteins whose entire job is to make cuts in DNA and to help relieve tension. They're called topoisomerases. What's important to note is that we're just going over these class of topoisomerases. Type 1 topoisomerase is not involved in replication and is not a gyrase. The next topoisomerase we talk about is. But topoisomerase type 1, the way in which it works is it cuts only one strand of the DNA, and it allows the remaining intact other strand of DNA to twist under the torsion and to help rejoin the nicked or cut strand of DNA. So it only helps to relieve the supercoiling. It only helps to relieve tension only by a little bit, because you're only cutting one strand and allowing the phosphate backbone to move a little bit of the DNA and then rejoin the nicked or cut strand of DNA. So because it does not alleviate supercoiling, this is not involved with replication and will not really address the supercoiling that occurs during replication. This is typically more present during transcription, where there's less opening of the DNA there. And there's no net energy expenditure here. It's and this is what it looks like with type 1 topoisomerase structurally, with the DNA present in the middle. The other class of topoisomerase is type 2. Type 2 topoisomerase is involved with replication and is typically referred to as a professional gyrase. What happens here is that there's a substrate or a site at which it functions. And again, it's isomerase, it's an enzyme. Substrate is what it's meant to target here. It's where DNA strands cross here. And so what it does is it creates a double-stranded cut in the DNA. And what it enables to do is it enables one strand to pass through the other and rejoin the cut strand. And so this uses energy, so it hydrolyzes an ATP. And what this actually looks like, so we're looking at a cross-section of type 2 topoisomerase, which is, again, a professional gyrase. We have a strand of DNA in green up top and type 2 topoisomerase depicted in blue here. So what's going to happen is that it's going to produce the cut of that DNA strand up top, and there's going to be another strand of DNA that has not been cut to be able to pass through. Okay, so again, we have our double-stranded cut, which enables another strand of DNA to pass through here. Sorry. So what's important to note again is that our prokaryotic DNA is circular. It's a circular genome. And at the once the entire template strand has been successfully replicated, the problem we're going to have is that it's basically two hoops linked together. Again, it's going to look like two magician's hoops that are stuck together. So there's going to be an issue that needs to be overcome of separating those two complete circular genomes in prokaryotic DNA. And so what ends up happening here is that type 2 topoisomerase is really critical for enabling the double-stranded break of one of the strands and enabling the passing through of the other. And it helps to disentangle these two complete genomes, enabling replication and further cell growth of prokaryotes here. So I'm going to stop here and ask, I'm going to stop for a couple questions here. Let me go back one slide. Yes.
So more generally, in there.
That's a completely different mechanism. Yeah. All right, we'll press on again. If you have another question, go ahead and shoot your hand up. So we've talked about the different, we've talked about a lot of the different important molecules that are involved with replication. One of the last really important molecules for us to discuss is DNA polymerase. DNA polymerase, we talked earlier about how elongation happens for the new strand in the 5' to 3' direction. And so that's the direction in which DNA polymerase functions. Some of the requirements for DNA polymerase to properly function is that it needs a template. It needs a template strand to read off of to produce a complementary and anti-parallel new strand. It also needs what's called deoxyribonucleoside triphosphates, which are DNTPs. Those are the nucleotides that actually have, usually there's a single phosphate group with each of them. Here we're going to have, it's called triphosphate, so there's going to be 3 phosphate groups here. What's important to note is that these DNTPs serve as both the source for the nucleotide, and so we have one DNTP here at the bottom where it provides the actual nucleotide that's going to be needed for elongation, but it's also going to provide the energy source, which is these 2 phosphate groups here. And so an important thing to note here again is that DNTPs provide both the nucleotide and the energy source for elongation. What's also important is that there needs to be a primer. Again, DNA polymerase, we talked about this really briefly, it can only elongate a DNA strand. It cannot initiate an entirely new DNA strand on its own. There needs to be a primer, something that you put in to get out more actual DNA strand. And so what that primer is actually is it is actually a small segment of RNA that's used within that's used to help serve as the primer. So it's mediated by a really unique molecule that's a DNA dependent RNA polymerase. So RNA polymerase, we typically think about it as making RNA during transcription. This is a really unique form of DNA dependent RNA polymerase that's called primase. So it's primase. It creates the RNA primer that's needed for replication to happen. So again, if I remember correctly, I think we'll have this on another slide. Nope. So what's important to note is that for DNA polymerase to work, there needs to be this RNA primer. And from that primer, DNA polymerase can then elongate off of it. Okay, we'll talk about how that RNA being present with our DNA is addressed. But the key takeaway from this slide is that primase produces this RNA primer that's needed so that DNA polymerase can actually elongate off of it. You can kind of think of, at least this is one thing that I found helpful. A good way to think about this, like you need the primer for DNA polymerase to work. Whenever you're painting your wall or painting your room, you need paint to paint. So here you need nucleotides to create new nucleotides. So you need the RNA nucleotides to create and grow more DNA nucleotides off of it. Yes.
Or is that
it's the initiator protein that identifies the origin. So I can go top to bottom real quick for the process. Initiator protein identifies the origin of replication. In eukaryotes, it can't melt. Prokaryotes, it can, but in eukaryotes, you need the helicase to come in and melt and separate the DNA. Single-stranded binding proteins will come in and stabilize it so that it doesn't re-anneal as it wants to do so because it's energetically favorable. You'll have gyrases on the ends, helping to address the supercoiling, making cuts and passing through DNA strands to make sure that the supercoiling isn't affecting the ability to then separate the DNA further downstream. And then what ends up happening is that then primase will now come in and lay down a really short RNA primer. Now that that primer is there at that origin of replication, DNA polymerase is going to come in and help promote elongation and create the new daughter strand. And the way in which DNA polymerase works here, we have two names for this class of DNA polymerase. It's DNA polymerase 3 in prokaryotes or DNA polymerase delta in eukaryotes. So we can call it DNA Pol 3 or DNA Pol delta. DNA Pol 3, I'll just call it that for now. Its polymerase function is in the 5' to 3' direction. The polymerase function is creating the new DNA growing strand. So DNA polymerase 3 here, or DNA Pol delta, will see the RNA primer that's been laid down by primase and will now carry out its polymerase function. It'll now lay down, using the DNTPs, those deoxyribonucleoside triphosphates, it'll lay down those new nucleotides that are complementary, but in an anti-parallel direction of the template strand. And so what's really important to note is that this process of adding these DNTPs, it's a really, it's the best way to describe is that there's a lot of sampling. There's a rich, high concentration of DNTPs within the nucleus. So there's this constant addition or adding and sampling of DNTPs at this specific base pair site where replication is happening. It's possible that the wrong base pair or the wrong nucleotide could be put in. If that happens, again, because we have very specific hydrogen bonding that happens between A's and T's and C's and G's, if that happens, there's an actual like physical disturbance or the wrong conformation. When that happens, DNA Pol 3 or DNA Pol delta can detect that disturbance or that weird conformation, and it will then do what's called a proofreading activity, or it'll actually go back in the opposite direction to do another function that's called serving as an exonuclease. Exo typically means at the ends. Nuclease just typically means removing the nucleotide. So it'll work backwards on the growing strand. So for example, here, as depicted on this figure, we'll envision there's a giant DNA Pol 3 or DNA Pol delta right here. The wrong nucleotide or a wrong base pair has been added here. This creates this weird conformation that the DNA polymerase can detect, DNA Pol 3 can detect. So it'll stop, lost my arrow. It'll stop adding in that direction once this wrong base pair has been added in, and it'll actually go back upwards. Now this is 3' to 5' to remove, that's the exonuclease, or proofread out that wrong base pair to allow for the right base pair to come in and bind. And so again, exonuclease means it's just removing the nucleotide at the ends. If this was an endonuclease, and we'll actually talk about endonucleases in another lecture, it's removing nucleotides in the middle of the DNA strand. And so again, this helps promote the complementary and anti-parallel DNA strand. That's the role of DNA Pol 3 or DNA Pol delta here. This is what it looks like with this DNA polymerase with DNA present within it. And this ability of DNA polymerase to bind to the DNA is really key, particularly binding and not letting go. Excuse me. And so this ability for DNA polymerase to bind and not let go is called the processivity clamp feature of DNA Pol 3 or DNA Pol delta. We have DNA Pol 3 depicted to the left here. That is a dimer. And then we have DNA Pol delta, the eukaryotic form, shown as a trimer. And so but they both share this overall structure that enables DNA Pol 3 or DNA Pol delta to bind to the DNA and not let go until replication is complete. I'm going to stop here for questions. Yeah, go ahead. Yeah, so the question is, does the proofreading activity happen after this, while the new strain is growing or after replication is done? It happens during. So if I go back a couple slides, you can envision in this image here that there's thousands of base pairs above and thousands of base pairs below. As the new daughter strand or the new strand is being synthesized, the proofreading or exonuclease activity of DNA Pol 3 or DNA Pol delta happens during replication. So anytime, and it's going to happen, anytime the wrong base pair is added, there's a difference in conformation that the polymerase can detect. So then it'll stop and go back up to remove that wrong nucleotide, enable the next DNDPs to start sampling. And once the right one is added there, then it'll continue on and keep going forward in a 5' to 3'. And so you can think of it as moving down in 5' to 3'. The wrong base pair is added, it'll stop, move up one, remove it, and try and enable the next one to be added. And if it's the right one, then it'll keep going and adding. And so like, if you envision it, it's kind of like this like back up and like moving down polymerase, exonuclease activity, polymerase, exonuclease activity, polymerase. So it's like generally moving this direction, but occasionally stopping if the wrong base pair is added there. Does that help clarify? Right.
The exonuclease, like up farther.
Yeah, so great question. So the question was, is the exonuclease a part of DNA Pol 3 or DNA Pol delta? We can think of what's depicted, what's listed here as different activities or functions that DNA Pol 3 or DNA Pol delta can do. It can serve as a polymerase in the 5' to 3', but it can also act as a exonuclease in the 3' to 5'. So the exonuclease activity is just for proofreading or correcting any wrong base pairs during replication. The main function is serving as a polymerase, adding nucleotides and creating this new growing strain of DNA. So these are just two different features of DNA Pol 3 or DNA Pol delta. Yeah.
But is this also?
Yes. And so we'll actually, I think we're 2 slides away from what that looks like. Hold that thought. All right, we have a few minutes. I think we can get to that slide. So I'm gonna go ahead and move forward. So we talked about the processivity clamp, the ability of it to, the importance of it binding and not letting go of the DNA during replication. So what this, again, so we talked about the replication bubble and the replication fork. What's really important to note here is that we have our primase that lays down this RNA primer, and then we have DNA Pol 3 that's now able to produce and create this new strand. What's important to note is that, as the question asked, this is happening on both strands that have been separated. So above, we also have, depicted on the bottom third here, we also have replication happening where there's an RNA primer that was laid down, and DNA Pol 3 or DNA Pol delta up top also growing and creating a new subsequent new strain of DNA on the top strain of DNA as well. What's important to note is that there's different nomenclature for these two new strands that are kind of being created at the same time. The bottom strand that's growing in the direction of the replication fork, we call that the leading strand, because that will be a contiguous strand of DNA that's growing, that's going to be synthesized and it's help us orient ourselves when it's growing in the direction of the replication fork. This will be referred to as a leading strand. What's important to note is that as this replication fork, or as this replication bubble is opened more and more to the right on this bottom third here, there's going to be more available DNA that can be replicated on the top. So what that's going to create is it's going to create these small fragments that'll then be synthesized or new replicated. That's the right phrase here to use here. New strands of the new strand will be replicated as more and more of this template strand becomes available as replication happens. And so again, when it's growing, when replication is happening in the direction of the replication fork, it'll be a contiguous strand. It doesn't need to be, it won't be these choppy strands, but it'll be contiguous, and that's the leading strand. And this will be the lagging strand. Again, because it's lagging because as the strands are opened more and more in this direction, more of it will be available to lay down a new RNA primer and elongate. New RNA primer and elongate. What's important to note is that, again, this is part of a replication bubble. That means there's a replication fork here, and there's going to be a replication fork, say, right here, which means when this top half is growing, it'll be growing in the direction of this replication fork. So that means on the other end of the replication fork, we have this, the top as the leading strand, and now the bottom will be the lagging strand. Because as we're opening in this direction, more sites for replication will become available as it's opened up. Does that help make sense how there's like a difference in leading strand and lagging strand on the different ends of the replication fork? Again, I'll try to get closer to the recording, sorry. Because as you're opening it up, more sites will be available here. So you'll have these short fragments, these are called Okazaki fragments, that'll be able to be replicated. But this will, because it's growing in the direction of the replication fork, as it continues to open up, it'll just continue going through. But it's almost the opposite on that end of the replication bubble. At the other replication fork, because as it's opened up over here, more sites will be available to replicate in that direction down here. But on the top, it'll grow towards that replication fork. So up top here is now the leading strand with reference to this replication fork on this side. Are there any questions about this slide? And it's okay, it always bears repeating. If something does not make sense, that's on me, not on you guys. So I will always accept that did not make sense. Yes. So the question is, if the DNA is being separated, the replication bubble is getting bigger. Won't it all be broken up fragments? Why is it contiguous? You can envision this is happening pretty fast. The ability of the DNA to be melted and DNA pol to go ahead and slide through and create DNA, like don't really think about it as traffic jam. This is happening pretty quickly, because also think about how quickly cells are constantly growing and dividing throughout our body. You can also think of tumor cells that grow rapidly fast. In some cases, not always, but typically we think of them as being cells that grow a ton. And so the ability to replicate DNA that's needed during cell growth or during mitosis is really important. So this isn't happening slowly where there's a traffic jam and I'm just kind of like waiting for this to open up and for it to go. It's happening pretty quickly. And so DNA pol 3 is flying down the template strand as this is opening up. As this is opening up, more segments are available here to then replicate. This is going, and once it hits here, it's hitting the other fragment and stopping. Once this area is available, it'll create it and hit there and stop. So that's why when you're growing away from the replication fork, you're only able to do it in fragments as more DNA, template DNA becomes available. When you're growing towards the replication fork, DNA is being separated and you're just flying through, creating the template strand. Does that help explain why it's contiguous at this point towards the replication bubble? It's not contiguous here, because this replication fork is making more available to grow in that direction as well. So there's going to be lots of this is not going to be one single contiguous fragment. When we look at it like this, it looks contiguous. But as we look at the other end of the replication bubble, there's going to be lots of fragments or Okazaki fragments here. Yeah. In the interest of time, last question, go ahead. Yeah, the question is, do the fragments, I'm going to reword your question. How do the fragments anneal together, how they join together? Stay tuned for next lecture. We'll talk about that. All right, with that, thanks for your time. Sorry for going a few minutes over.